Welcome to the Minute Science channel. Can the DNA be changed easily? Can alterations be made to it? Can the humans get rid of thousands of genetic diseases that have plagued mankind from the dawn of time? The question arises, can the DNA be edited? And if you're asking yourself this question, you're right, it can be. But it is done in a laboratory environment. It is edited using highly specialized chemicals, which make additions or deletions of genes at very specific sites inside the DNA. Actually, the DNA editing process is much like the video production process. Like in video production, we have to make cuts to make the final scene look good. The DNA can also be changed and edited by using highly specialized scissors, which are called as the molecular scissors. And these molecular scissors work automatically. No manual intervention is required in the process. We can't manually cut the DNA because it is very small. It is a teeny tiny molecule inside the cell. So to cut the specific sites, we use these molecular scissors, which make precise cuts inside the DNA. Earlier, the DNA editing process was very daunting. It was hard to perform. It took months and years to perfect. It was very time consuming, and in that time, only small changes were made inside the DNA. It was costly because the chemicals used were very costly, and the process was also error prone. It would target any other site or would have not made the required changes. So I bring you CRISPR, also called as the clustered, regularly interspaced, short palindromic repeats. Let's make it short for CRISPR. The technique is not so hard to understand. It is a new age technology of gene editing. This technology is easy to perform. It is fast as compared to the previous technologies. It does not take months or years to perfect. The gene can be edited in a few weeks' time. It is also cheap because the chemicals that are used are very easily extracted from the bacterial cells. So where this technique called as CRISPR originated? Actually, the bacteria has its own immune system. It is similar to the immune system that the humans have. It protects the bacteria from invaders, and the bacteria has bacteriophages, or they are the kind of viruses that infect bacteria. Whenever a virus infects the bacteria, it does so by unpacking itself and injecting a small viral DNA inside the bacterial cell, and this viral DNA then makes copies of itself and new viruses are produced. But the bacterial immune system slices this viral DNA and keeps a short part of the viral DNA with itself. It stores it inside the bacterial genome or the bacterial DNA. The site where the viral genome is stored is kind of an archive where the viral genome is kept locked inside, safe for future use. It acts as a memory for the immune system of the bacteria. The part of the viral genome is stored inside the bacterial DNA in a CRISPR gene. This CRISPR site has repeats which are interleaved by spacer DNA, and each spacer DNA is unique because it codes for different viruses that have infected the bacteria previously, so it acts as a memory for the cell. So during the subsequent infection, when the virus infects the bacteria, the immune system is triggered. The spacer, which holds the copy of the segment of the viral DNA, forms an RNA. So when the RNA binds to a protein that is called as Cas protein, this Cas protein is a highly specialized protein inside the bacteria. It is designed to cut the DNA whenever it binds to RNA, and Cas stands for CRISPR associated proteins. So this Cas9 protein is a specialized DNA cutting protein, and all the DNA cutting proteins are called as nucleases. It is kind of an endonuclease which cuts the DNA from inside. So whenever a virus enters inside the bacterial cell, it releases its genome inside the cell, and the bacterial immune system gets activated, and an RNA molecule specific to that virus is produced by the specific spacer that is present at the CRISPR locus, and this RNA then binds to the Cas9 protein inside the bacterial cell. So this RNA is called as guide RNA, as it guides the Cas9 protein towards the viral genome. And when this Cas9 proteins encounters the specific viral genome inside the bacterial cell, it acts as a nuclease, a molecular scissor, and cuts the viral genome into two halves and renders it ineffective. The Cas9 cuts the viral genome inside the bacteria by attacking at the target site that is similar to the sequence of guide RNA. This property of Cas9 linking with the RNA provides immunity to the bacterial cells against viruses. And this CRISPR technology can be used for gene editing purposes, where the Cas9 protein can be extracted from the bacteria. So when the Cas9 protein combined with the RNA that is complementary to the gene of interest we have to cut, it specifically searches for that site inside the genome, and when it finds the site, it makes the necessary edits to it. So whenever the target site is found on the genome, the Cas9 protein cuts the site. And Cas9 is special because it cuts both the strands of DNA, so it separates the two DNA fragments at the cut site. So when the Cas9 cuts the DNA at its two strands, the cell thinks that the DNA has been damaged and tries to repair the DNA. And the repair process of DNA starts inside the cell. So there are two processes by which the DNA can be repaired inside the cell. The first process is stitching back the damaged DNA. So this is done where the damaged DNA is stitched back by some enzymes inside the cell. But these enzymes are not very efficient in nature. They remove some of the nucleotides that are present at the cut ends of the DNA and tries to stitch back together the rest of the DNA. So in this process, some of the nucleotides are removed and the rest is joined back, and this results in loss of gene at the target site, and the target genes becomes inactive in the process. In the second process, where a new gene can be inserted at the target site, in this process the gene of interest is added inside the cell, where the DNA repair mechanism inserts it at the target site. So when the DNA gets damaged, and if we provide the gene of interest inside the cell, the DNA repair mechanism will insert that segment or that gene at the target site through the DNA repair process, and this is called as insertion, and it can be used to insert new genes inside the DNA. What are the uses of this CRISPR technology? It can be used to add or delete gene at a very specific target site inside the human genome. Genes can also be switched on or off to observe their function. By this CRISPR technology, the genetic disorders can be treated very swiftly because the CRISPR technology can be performed in a few weeks' time, which is very fast and can work on adult human cells also. Creation of designer babies if gene editing is performed earlier at the embryonic stage. If the change is made to the embryo, the result is observed in all the cells of the body. Please like my video if you like this content. Share it among your friends and family to let them know about the gene editing process, and do subscribe to my channel for more videos in the future.